Hello guys, welcome back to my channel. If you're an old subscriber, but if you're a new subscriber and you're just seeing this channel for the first time, please do well to subscribe. So in this video, we'll be talking about the biology waec specimen for 2025. So let's get to it. All right, so specimen A is the bean weevil. And the most important thing you need to know about this specimen is the scientific name, the kingdom it falls under, the phylum, and the class. So the scientific name of bean weevil is Acanthocelides obstectus. So that's the scientific name. So it falls under kingdom Animalia, phylum Arthropoda, which is the largest phylum, and class Insecta. Now, one of the likely questions you should expect is to list observable features. This is very important. You look at the specimen, you observe it. Now, what are the observable features of um, the weevil? First, the specimen is divided into three segments. Normally insects are divided into three segments. So we have the head region, the thorax, and the abdomen. That's the first observable feature you can note. Secondly, it has three pairs of legs. Okay, three pairs of legs, or you can say three pairs of jointed appendages. So you can see the legs there, three pairs. Thirdly, presence of compound eyes. So the weevil has the compound eyes. So number four, typically small in size. Remember, we're talking about observable feature, what you can observe while looking at the specimen. So it's typically small in size. It is oval in shape and dark brown to black color, depending on what you see. So this is one of the questions that can come out. You can also be asked to list the economic importance of weevil. Now, when we talk about economic importance, we are not only talking about the positive importance. As a matter of fact, it is actually the negative impact it has on the economy. Okay, so number one is that it causes or it causes severe damage to crop. Number two, it can reduce crop yield. Three, it leads to food insecurity or scarcity. So when this weevil starts pestering on the beans, right, it can reduce the yield of beans. So that's what that means. And number four, infestation can cause scarcity and escalate price of beans. So the more infestation, the more scarcity, and the more hike in price. So these are the economic importance in case you are asked to list them. All right, so for other questions that can emanate from this particular specimen is the relationship between specimen A and B because they are related. They can ask you what's the relationship between specimen A and B, and that's very simple. Specimen A, which is weevil, infects specimen B. So specimen A feed on specimen B. So they are related. So that's the relationship between specimen A and specimen B. It feeds on specimen A, B rather. It derives shelter from specimen B. It derives nutrients from specimen B. So it's basically a parasitic lifestyle. Okay, so it parasitizes on the bean seed, or you can say the bean plant. All right, so another question that can come out is draw specimen A. They can ask you to draw the specimen. Now, this is very important. I often tell my students, draw what you see, don't draw what you have in your head. Now, don't look at this diagram on this video now and imagine that that's what it should look like. No, draw what you have on your table. So whatever is presented for you on your table, make sure you draw it, because the truth is that your teachers will be asked to list out the things that were provided for you. So if you draw what you have in your head and not what is on the table, meanwhile your teacher has already given them what he or she provided, that means you have failed that particular question. So draw what you see. Draw what you see. Very important. Label and title appropriately. Don't forget to write your magnification. That's very important. So you put your labels, your title, and your magnification just by the right side, below your diagram. These three things are more important than your diagram. Please don't forget that. Now, other questions that can come out is adaptive features. They have hard exoskeleton, and they have chewing mouth parts. So these are features that can help them adapt to parasitic lifestyle as well. So specimen B is bean seed with holes. Now, looking at the specimen, the botanical name of bean, you must know this one, is Phaseolus vulgaris. Very important. It's under kingdom Plantae. So you may be asked the observable features. Now, the first observable feature you can see right in front of you is that there are holes on the surface of the specimen. Okay, there are holes. Secondly, obviously there will be powdery residue. So you see um things like powder. So when you touch the specimen, you feel powdery substances. Okay. Now you can find discoloration from white to maybe black or brown as a result of the infestation. You can also find weak texture. The bean can become very weak. So these are observable features. You may also be asked what you can do to prevent the spoilage of specimen B. Now, how do you prevent the spoilage of specimen B? Firstly, store in a cool, dry place to prevent mold growth. Secondly, store in airtight containers to prevent insect infestation so that these insects wouldn't reach it. Thirdly, avoid storing in porous or open bag. Porous means bags with holes. Don't store beans in bags with holes or the bags that are open. Okay. Thirdly, control pests by using natural repellents like bay leaf. There are some leaves that are natural repellents. They repel what's it called? They repel the weevil, right? Or pests or any pest that wants to come close to it. So some natural repellents like bay leaf or neem leaf as well can be used to repel these pests. Fourthly, fifthly, check stored beans regularly for spoilage. So when you store your beans, check them regularly. If there's anyone that um, is bad, you take it off so it doesn't infect others. So these are ways to prevent the spoilage of specimen B. Other questions can include what type of germination does specimen B exhibit? So because it is a dicot plant, it will exhibit what? Epigeo germination. Okay, so this specimen exhibits epigeo germination. They can ask you what part of the specimen stores food. It is the cotyledon. So the cotyledon is a part of the specimen that stores food. You should also know that it has an annual life cycle. That means it completes its life cycle in one planting season. Okay, so those are the questions you should expect from this specimen. So specimen C is water leaf. So what are the questions we should expect? Firstly, you should know the botanical name of specimen C, and it is Talinum fruticosum. So that's the botanical name of water leaf. Now it's under kingdom Plantae. So what are the characteristics of this specimen? They have flowers, they have cylindrical stem, it has shallow root system. So if you uproot it, because I think this specimen you're supposed to be presented with a whole water leaf, so both the leaves, the flowers, and the roots. So it has a very shallow root system. So that's that. Other questions include draw a leaf from the specimen. So you can be asked to draw a leaf. So what you just do is pluck out a leaf, place it in front of you, and draw what you see. Draw what you see, label appropriately, um, put your title, very important. Title is even more important. So put your title there and put your magnification. How do you get your magnification? The magnification is the size of your live specimen divided by the size of your drawing. So measure your drawing you have on your in your answer booklet, then measure your live specimen, then divide the value, you would get your magnification. Write it by the side of your diagram, the lower right side of your diagram. So specimen D is quill feathers. What do you need to know about this specimen? Firstly, The question can come in this manner: what is the kingdom and class from which specimen D can be obtained? So it comes from kingdom Animalia and phylum Aves. Okay, kingdom Animalia and phylum Aves. Now you look at the location on the body of organisms. It can be found on the wings or the tail of the bird or the domestic fowl. An example of organisms that is associated with this specimen, we have the domestic fowl, the pigeon, the turkey, and so on. You might also be asked to draw the specimen. So if you are drawing the specimen, this is what your drawing should look like. You should label the quill, the after shaft, the barbs, the shaft, and the veins. Draw what you can see on your table. Write your title, very important. Put your labeling and your magnification. Don't ever forget magnification in practicals. It is very important. So you must include the magnification, your title, and your label. Specimen E is pineapple. Pineapple is specimen E, longitudinal section. So that simply means we are going to cut the specimen from up to down. When you say longitudinal, it is from up to down. So the first thing you need to know is the botanical name of specimen E, which is pineapple. It is called Ananas commosus. Ananas commosus. So that's the botanical name. The kingdom Plantae, division Magnoliophyta. So when we talk about plants, plants don't have phylum. Rather, we call them divisions. Okay, so the type of fruit it is. So pineapple is a multiple fruit and it developed from an inferior ovary. So the ovary type is inferior ovary. So in case you are asked what ovary type is specimen E, so it is inferior ovary. You can also be asked to draw the specimen. So if you're asked to draw the specimen, this is what your drawing should look like. Although this is not a live drawing, this is not a paper drawing, this is a live specimen. So you draw exactly what you see. So you have your main labeling, the crown, the base of the crown, the shell, this is the outer part of the pineapple, the ovary, the sepal and bract bases. Then we have the inflorescence axis, that's the core. The inflorescence axis, that's the pillar or the core of the fruit. Then we have the cortex of the core, and the base of the specimen. So these are what you should take note of. So the next specimen is specimen F, which is orange fruit. Now in this case, we are asked to do a transverse section of the orange fruit. So TS, transverse section. When we talk about transverse section, you're cutting the specimen from left to right or right to left. So what do you need to know about the specimen? Botanical name, Citrus sinensis, very simple. Under kingdom Plantae, division Trachophyta. Very importantly, you need to know the placentation of orange fruit. So the placentation of orange fruit is axial placentation. Axial placentation. What type of germination does this specimen exhibit? In case you are asked, it exhibits the epigynous germination. Please note this. And in case you're asked to draw the specimen, this is what your drawing should look like. Exactly. This is a pencil drawing. So this is what your drawing should look like. Draw what you can see. So the drawing has very important labels: the epicap, the mesocap, and the endocap. Very important. Then you go over to labeling the seed, the placenta, and the succulent hairs or the succulent part. Most importantly, don't forget your title. Your title is very important, and also include your magnification. You measure the specimen on your book, measure the specimen, the live specimen, divide the values, whatever you get, include it in your magnification. So that is what you should do for this particular specimen if you are asked to draw. All right, so we'll be treating specimen G, which is raw egg of domestic fowl, and specimen H, which is test tube containing 3 ml of water in a rack. So it's expected that your teachers will provide, or your teacher will provide, the test tube that contains 3 mils of water, and raw egg as specimen G and H, respectively. So, in these particular specimens, what we can say that will come out, or what we can assume you will see, is just the normal burette test, chemical test for protein. How do you test for protein? And I think you would use the sodium hydroxide and the 1% copper sulfate, yes, to carry out this experiment. So just note this. You should note the outcome already, but you have to carry out the experiment there so that you will see your outcome and record. I think you have to record in um, test, observation, and inference boxes. So for your test, you just write your test for protein or burette test, your inference, your observation will be what you observe. What color did it change to? Did you see precipitation? Did you see bubbles? Whatever the whatever you observe there, you write it in your observation box. Then move to your inference. Inference is your judgment. What is there? So inference is protein present or protein absent based on your work, based on what you have done. So that is what you should expect with specimen G and H. And finally, specimen J, which is longitudinal section of boiled egg. So you would see on that day on your table, you will see boiled egg, longitudinal section. So it was cut up down. So the picture you can see by the side is what you will be expecting, what you should expect to see. Now, what are the questions, likely questions that you might see? They might ask you to mention the parts and the functions of the parts. Okay, so you have the albumin. What does the albumin do? It supplies protein. The yolk, what does it do? Provides food for embryonic growth and development, or the growth of the embryo. Thirdly, the shell. What does the shell do? Protection, aeration, shape, gaseous exchange. The shell membrane also allows gaseous exchange. So you have the cell membrane, the shell membrane rather, before you have the main shell. So these are likely questions that you should expect from specimen J. You can also be asked the economic importance of specimen A, or what is the nutritional benefit of specimen A, of specimen J rather. Of course, it is um a proteinous food. It is high in protein, high in the 9 amino acids. So these are the things you should note in um specimen J. You can also ask you what animal is associated with specimen J. You know it is the domestic fowl, all right, or the bird. Now you can also be asked to draw the specimen. So if you're asked to draw the specimen, this is what your drawing should look like. You have the shell membrane. The shell is the outer part, then the shell membrane is the part that follows the outer part. Then you have the chorion, and your yolk, your egg white, which is also called the albumin, then your S space, that is if you can find an S space there. So this is what you should expect.